Good to go. Awesome. Great. Well, I'm really excited to have Dr. Chen Wang here. He is at Mayo Clinic, and he is a really accomplished bioinformatician and researcher, and has become a good colleague and friend of mine here at Mayo Clinic. And we get together and we chat about things like complex variants, which is what you're going to hear about today. So without further ado, I'll hand it over to you, Chen, and then yeah, we can kind of collaborate on this.
Yeah, hello everyone. I think like I don't have many slides. Probably let's turn into interactive discussion because this is an important topic for several of us. Probably I should do a brief self-introduction. My name is Chen. I'm a bioinformatician, joined Mayo Clinic for 10 years. I guess my past experience on this matter of subject is more coming from TCGA, the Cancer Genome Atlas. I was with uterine cancer and recent liver cancer and a bunch of pan cancer integration analysis. Complex variants such as copy number variation or the fusion or structure variant are also like intimidating topic. The other side of my background is working with the clinical genetics lab here. It's like offer the clinical or commercial grade of the result to patient and physician. And structure variant or complex variant, how to report, how to exchange, even between different institutions become such a challenge. So with that, like maybe I will just quickly go through a few slides, not too many. Hopefully we'll have more than enough time to discuss. I think we were aware of like in the human genome, there are complex variants beyond single nucleotide change, SNV or SNP. That it could be deletion, insertion, duplication, inversion, translocation, etc. And the structure variants could happen in DNA level, or like you could detect it in the RNA level as a fusion. For example, sometimes deletion between two genes could introduce like a de novo fusion. And not to mention, like I was thinking, the recent interesting topic from cell, like there was like fungus or virus insertion. So the complex variant to me, it could be, you know, just different chunk of human genome they fuse together, or it could be virus or like there's like virus-induced oncogenetic event. But come to like a bioinformatics, and especially, you know, if we think about clinical usage of the structure variant CNV, I think relatively speaking, we lack the common data model to do the representation. Although in general, you can always describe the chromosome location, but there are like a different terminology according to the file principle, like findable, accessible, interoperable, and reusable. Because I think in the next few slides, maybe I have more like a dense information enriched example. The left was a cytogenetic example. It was so fun, was provided by Beth several months ago to me. Like when you find a complex event, like a gene fusion and maybe companioned by many other structure variant event. If you are not from cytogenetic background, it will become like foreign language to you. Whereas if we are bioinformatician, maybe VICC format on the right is what we are familiar with. But either way, I can say that's a gap, you know, how we represent a complex, simple to complex structure variant. How do we make it computable? How do we make like, you know, interoperable, you know, between one study and another study? And this is like simple Google Scholar search. You certainly can do according to your favorite structure variant or copy number caller. I think they will have diverse output format. Last week or the week before, I had a conversation with another colleague of mine at Oregon Health. Like for TCG, he hosted a Fusion Dream Challenge several years ago. he told me even imported data like from different SV or fusion caller was the #1 challenge, right? Because each caller will give you convenient but different diverse set of the results. And like from each of the TCG studies, it's always amazing to me, like at least the copy number or the recurrent copy number change will be one of the figure. For example, this is the ovarian cancer, one of my research area of interest. You can see there are recurrent focal amplification around the VICC and a recurrent focal deletion of tumor suppressor gene region. But if we don't have a good data representation model, it's very hard to capture such knowledge and incorporate into the future like a clinical either research or practice. That's one of the motivation for us to discuss with several research group, include VICC here. And some of the CNV, like the global pattern of CNV, this is from the uterine cancer. As you can see, there are 4 groups of copy number variation patient across, I guess at that time, 300 patients. They have tremendous significant association with outcome. Like the green group of patients, these are the uterine cancer, they are copy number quiet. Because basically this group of patients, they are the MSI or POE, ultra or hyper mutated tumor. And their copy number is quiet, and their outcome is great, versus the very right group. These are the genome unstable tumors globally, and therefore like their tumor probably is the most aggressive and outcome is the worst. And such kind of study or association, this is just like one instance. Probably you can say from here and there individual disease or TCG study, but it's never easy to capture to say, okay, copy number quiet or genome instability high, like in a computable way. Again, like I'm arguing the necessity or motivation to having a data representation model, not only for the data representation, but as well to capture the knowledge relationship, including the clinical outcome. As I briefly mentioned, like the other head of me was working with the liver cancer group here as liver cancer spore, the specialized disease center bioinformatics core. Some particular structure variants, they are implicated in the diagnosis of certain tumor. Like this is one of the very intriguing real, actually it's like the liver cancer only occur in young adult or kids. I was a little hesitate to say the pediatric cancer because it can also occur in the younger adult. But basically the deletion, it's 100% recurrent deletion between these 2 genes, PRKCA and the DNA JB1. And the result of DNA level genomic deletion will introduce de novo, its oncogenetic fusion of these 2 transcripts into a new gene. And this fusion itself conclude the diagnosis for this rare liver cancer. It was so amazing, published like 12 years ago in Science. It's 100% recurrence. And the diagnosis test actually is developed here in Mayo Clinical. They use the fish, was so simple. But such kind of like, important, rare, but clinical important, structure variant change, only if we can calculate computationally and data representation, then we can trigger like even the clinical decision support. And by the way, if you search these fusion in CBI portal, like what the cases you find will be from this fibrilloma liver cancer. It's a diagnosis implicated. So and through the years, the technology of the sequencing, early on, the detection of CNV, SV, or especially copy number variation depends on microarray. Then the sequencing, the short reads, next generation sequencing come to the stage. And nowadays, I believe you hear that like the long reads, nanopore, PacBio, we are also overwhelmed by the next generation new technology to measure structure and variants. And therefore, even the metadata model, like describe a set of variants, they were detected from microarray from array CGH or SNP microarray or like Angular 2 channel versus short arrays, nanopore, pack-by long arrays become a necessity, because otherwise, even if we can represent the data, we don't know where the data coming from what kind of study, and from what kind of a device. So I can say, like, we also have the a challenge to motivate us to discuss like the metadata, like what will be the required metadata to describe CNV SV complex variant study to make the dataset reusable. Because you cannot necessarily compare microarray-based versus NGS-based study. One can reach base pair level resolution, the other have some kind of gap area between different microarray probe. With that, like Beth and I already have a few rounds of discussion. We really want to approach different like a working group or consortium, but in the way that can we get a focus group to tackle like these emerging need for representing the complex structure variant, including CNV, like a genomic level CNV and ion level fusion. Yeah, and personally, I'm associated with AMIA, American Medical Informatics Association, for 5 or 6 years, and more closely with translation bioinformatics group. Certainly, I was like growing up in TCGA for 7 or 8 years, well aware of like the research opportunity, but also like information exchange challenge over there. And Beth was recommend me to like get in touch with the VICC group, knowing that this group is very on top of like oncogenetic variants and could be interested in this topic. We are also talking about the possibility to forming such a focus working group in CGC, because many of the like initial discovery and usage of the complex structure variants is coming from clinical side, as the like young adult / pediatric cancer situation I refer to. And also just have a little bit expected outcome from such working group. We are hoping not immediately getting like a perfect data model or metadata model, rather than being able to engage with different player in the field, because even structural variant, it could be detected by informatician like me, right? Run a bunch of caller, either VCF or arbitrary beta file tool, but it will be like annotated or consumed by clinical genetics people that they are aware of, like these are polymorphism, it's pathogenicity, and could be associated with some disease condition. And also we have the like data modeling people from GA4GH or HL7. So certainly it's very complex topic, would require multiple party of, people to join. But hopefully this group start like, yeah, my slides are very short in the way that I don't want to occupy too much time, talk about my own work rather than just talk about challenge and engage with your question or comment.
Yeah, thanks, Chen. I just wanted to kind of highlight that mechanism that you brought up with the deletion between DNA JB1 and PRK ACA. I mean, a lot of us are very familiar with mechanisms like this, where a deletion can be seen by one methodology and it results in a fusion. I mean, those who work with brain tumors are very aware of the BRAF KIA 1549 fusion that's seen as a CNV on microarray. And we actually call fusions based on that CNV detection by microarray, which is not a breakpoint specific assay. And that fusion is associated with pilocytic astrocytoma. So, I mean, this mechanism, which is really hard to describe with our current kind of annotation and representation, it's rather common in cancer. So I think it's a really relevant and important topic.
Yeah, it's a common overall in cancer, but in this particular, it's a rare liver cancer. I mean, by the way, like maybe the story of this science paper, the last author is the father of the daughter of one of the co-first author. She's diagnosed with their rare cancer, and the father like dedicated entire his career to study this cancer and establish the clinical like a registry across the world. But it's a small instance. I always imagine a situation when, you know, maybe you or me or like from some clinical lab, they detect this fusion without some computable knowledge source. They may let it go. They may not aware of like it could be diagnosis implicated event. And moving forward with such high specificity of the event, they develop multiple assays, not only for the tumor for FIS, they also develop for the serum, like to monitor early recurrence of this tumor. So being able to describe these events unambiguously, I think lay out such an important foundation for the future, like reuse, clinical decision support, etc.
Completely agree. So I guess my question, I mean, some of you are aware, we did have a table discussion at CGC based on kind of complex variants. And we started with the question, well, what is a complex variant? Because it could mean different things to different people. We kind of landed on, you know, not your typical SNV. So it could be a CNV, could be a fusion, could be, you know, a co-occurring mutations, things that kind of fall outside of that single nucleotide variant or kind of simple mutation. And I think that what you're presenting here is very much in line with that. And yeah, so I think that's really good. I know that we do have interest in the CGC group, but also I wanted to see if there's interest in the VICC group as well, since this group does represent a lot of other skill bases as well with bioinformatics, and wondering if we could kind of bridge that gap between kind of the clinical laboratory experts and informatics to kind of gain some traction here. So I'm kind of wondering what opinions of those on the call are about kind of moving forward and proposing a working group for this.
I can contribute my thoughts. You know, I think the notion of a structural variation, you know, focus group makes a lot of sense. You know, this was for those of you that were at the GA4GH plenary, which was a couple of weeks ago. You know, one of the big discussion points that came up during the GKS workstream call was, you know, this notion of SVs and SV representation. And so I think that there's a pretty strong community interest in being able to represent these in a reproducible way. I wanted to comment on something Dr. Wang mentioned regarding like assay metadata, which I thought was really spot on regarding like how do we represent you know, where the data come from, because I think that's valid. It's a valid, you know, piece of information in terms of, you know, interpreting the variation concept. And, you know, in the gene fusion group in VICC, you know, we basically had this metadata field, and we ended up using the ontology of biomedical investigations and using the structured terms there to represent the various assays like FISH or high throughput sequencing, et cetera. I was wondering, Dr. Wang, if you've looked into ontologies or systems for representing this, and if you have any recommendations of your own regarding like resources for, you know, computably identifying where the data came from.
Yes, thank you. Could you put that link into the chat? I think that would be very interesting. Yeah, because early day, back to you, Alex. I mean, early day, think about microarray. They have the minimum like a data representation schema, that was from like for GEO, right? So definitely when you talk about ontology, I was trying to be very cautious, not reinvent the wheel, rather than borrow what's already available. There are two types of ontology. One is for representing the structure variant itself. For example, you may have a copy number neutral and a copy number like a dosage change, right? That's different ontology branch, but you more refer to the metadata. And I think the metadata, definitely we can follow some existing schema from like a microarray, the minimum data element. It's called a minimum or something like that. And also for SRA, right? Because we have the sequencing archive right now, they also have their metadata. But unfortunately, the reality is if you look into some geo data set, they have the required field, but there's no such like a compliance check or like a data element check. Many of them are missing or like a typo. So kind of they have the recommendation, but they don't have the tool to encourage with the community adoption or help them with like checking of consistency. Certainly there are, and these are like a legacy from microarray and short arrays. Coming from the long arrays, this even become harder. And talk about structure variant per se, often when we like start with our research trying to reuse, the difficulty is coming from like whether people like describe single end, pair ends, and when they describe the calling, the base pair resolution, like what's the buffer room? Like, you know, are they really after the base pair resolution or they have some wiggle room because of the caller? So some of metadata actually is also associated with the caller, like how confident and at what resolution you call the structure variant. But that's a great question. Like we're trying to be smart, not to reinvent the wheel too much. We also want to specify the requirement. For me, let's say compare with another American study of like a breaking point happening in another population. You know, am I really clear at what resolution and what technology they use? So I believe or agree or disagree with their conclusion, you know. Yeah, thank you.
Oh, I think that's a really good point. And then figuring out how to annotate those different resolutions is another challenge that we absolutely do have, because with sequencing assays, often we're down to the exact base pair. With RNA-seq, we're down to exon most of the time. With microarray, we're limited to where the probes are in the assay. And with FISH, the resolution is even you know, lower. So it's a really interesting and kind of cool concept.
Yeah, I mean, just to maybe conclude, it's just a supplement what Alex mentioned. I think metadata really should be described as to study metadata, right? Like, are we doing the case control and from tissue or blood? This study metadata. Then we have the platform metadata. Is this RNA sequencing? But if the RNA sequencing is poly A selection versus RNA capture, right? It's another difference that will lead to the bias where you can capture or, you know, detect the fusion. And the third aspect of metadata, very often, I'm not sure it's totally ignored, but could be overlooked, which is the metadata associated with the bioinformatics caller. Because it depends on which caller, which version, and what intermediate result you generated. That's a gap that you may or may not capture the entire spectrum information. So you'll be, I mean, hopefully you'll join our working group to be formed. We need to detail the type of metadata, different category.
Cool, really good discussion. Are there any other thoughts or questions on this, or any other comments on, you know, interest in this group, or any knowledge of groups doing similar things like that?
I have one follow-up question about CNVs. One of the big challenges with CNVs that we ran into when modeling this for the variation representation specification was the notion of kind of like the like absolute quantitative count of copies as called versus kind of relative metrics, you know, low level gain, high level gain, copy neutral, you know. I think it'd be, and I know that various efforts over the years have like tried to do this, but it'd be really nice if there was more of a formalization or community consensus around certain terms like amplification and what that means.
Right.
And you know, especially when you're trying to describe like, you know, these relative events, and often we have to do that in cancers because of the complexities of the samples that we're looking at. So I'd like to hear your thoughts on relative copy number expressions.
Yeah, Alex, if I can add to that, we struggle a lot with like deletion versus loss. What does that mean? Like when is it a, how do you know, like how do you standardize on whether it's a full gene deletion or an exonic deletion defined by certain, you know, or can loss be full gene or hemizygous, or, you know, just a couple of exon loss? Like how are you defining loss versus deletion is a big one. And also other structural variants with respect to genes. Are you losing, what are you sequencing? What do you still see? Do you see the 5' end or the 3' end of the gene? Where's the break point? Is it do you call it a truncation or a rearrangement or, you know, or something else?
Right.
It's not at all standardized.
If I can try to respond first.
Go ahead.
This is a fantastic question because like we all struggle with enjoying this question. I think the example I give out was like using Portal. Like Portal, according to TCGA, you have the raw data, right? You have the intermediate, like normalized data. Then you have the core data. So back to Alex's comments, like copy number is dosage data. So you have measurements after, you know, very magic normalization and contrast by 2-channel or virtual channel pool of normal sample. You have the relative log to ratio. That's like a level 2. Like it's objective quantifiable signal that you believe is like 1 copy or half copy more, but it's not incorporated into the per sample like a purity or like overall ploidy to core your copy number. And to core the copy number is like a genotyping. It itself is inference, has inference like accuracy limitation. So like according to the other question, right? Like you talk about the loss versus deletion. Sometimes I feel it's the technical that hopefully the cytogenetic folks can work with us to define. At the beginning, I'm into this field. I ask exactly the same question. Like how many copy you call as amplification? They say like it could be 10 or 20, like 3 or 4 often we call as gain, you know. Like hopefully we have some clinical cytogenetic, if not like a best, just bring some people in to resolve some of terminology like that. On the other hand, like loss versus deletion, yeah, I think exactly is, you know, if it's 1 copy, we call loss, right? If both copy call deletion. On the other hand, it could become so complex if we get into the purity or clonality issue. So like this is the level 3 or 4. You integrate all the intermediate or normalized data or information, trying to do the best inference, like a genotype state of that. So it will be serious of like a representation requirement from how we represent the metadata intermediate signal versus to the inferred result. And the inferred result often is condition on caller as well. Thank you. I mean, I'm not sure address any question, just like my experience.
So I see we have a couple people with their hands raised.
I'm not sure. Yes, this is Gordon. I'm not sure if you might be, if you are aware of this, but the standards that were developed through collaboration between ACMG, LabQA, Cytogenetics Subcommittee, and CGC try to define some of these terms, amplification, gain, loss, and because there was awareness, there has been awareness for a long time that these are not being always consistently used. So that's a starting point. I'm not saying that there is no more work needed, but at least we have some starting point there. They offer some definitions. And when it's considered amplification versus just gain, what is kind of a threshold, And that's their suggestion, where we may or may not agree with. In terms of whether to call something loss gain versus deletion, that same manuscript recommends gain and loss as more broader terms. In a loss situation, deletion and loss may be interchangeable, may mean the same things, but calling something duplication is a dangerous approach, right? Because not all gains are duplications. They could be insertions. It could be duplication means something usually like in situ. So as general rule, that same manuscript recommends gain and loss rather than duplication deletion. So just wanted to make everybody aware that particular manuscript starts to address some of those questions, and further work might be needed, but maybe we can build on that rather than starting from scratch.
Yeah, thank you very much. That's very important information. As I mentioned, like we're trying to be mindful not to reinvent the wheel. Thank you for many of your comments. Indeed, like amplification could be like amplify and insert somewhere else. So although dosage wise is like a multiple copy, but actually structural variant could have other implications. Thank you.
Yeah, I wanted to say again on amplification front that it's not necessarily or not always the only criteria is the number of copies, right? Because amplification has a different nature as compared to gain, and amplification it's a focal event versus say arm level event, which
Exactly.
Would be
Exactly,
gain.
that's what I'm saying. The definition might not be complete that they're providing in that manuscript, and it should be probably built in that. Mechanism is also probably different between amplification and just gain. Excellent point. So I think we still have work to do, but maybe, as I said, not starting from scratch and
Yeah,
probably not contradicting what's already out there.
No, we shouldn't start from scratch. I mean, there's a ample literature, and it would be a good idea to have in a working group people who already been kind of involved in some of those discussions and have clinical experience on what they call amplifications and gains and homozygous and heterozygous deletions.
Yeah, and also, I mean, when we talk about amplification, I think the most immediate example I can recall is HER2, right? Breast HER2. If it's like IHC versus fish, and we have amplification and even the borderline situation, like how do we just call it? Just in the between, you are not clearly saying that's amplified or not. And it could also differ between the technology. That's maybe a call for metadata as well. But if we measure the same HER2 tumor using whole genome sequencing, then you may get a slightly different or high resolution to describe that structure variant.
Right. And then on top of that, I mean, I think that a lot of these caveats have been brought up already, but I mean, in real clinical lab practice, a lot of these specifications on what is an amplification can be gene specific. So, our threshold for calling a ERBB2 amplification is much lower than some other genes that we would expect to be amplified at a much higher rate. And so it's a really confusing topic.
Yeah, I think it's a rabbit hole, right? You get into ERBB, HER2, MYC, EGFR, like, could be people, like, depending on the clinical domain you are in, you have different expectations. But back to the informatic aspect, if we can represent amplification as objective as the initial relative dosage, log2 ratio, and the companion, like deploy or apply the evidence, that can be done as a foundation, not immediately getting into conclusion, right? Yeah, but good comment and good question. What I heard is like, I mean, there's a slew of works like already, doing and trying to clarify the definition over there. We would love to, you'll be able to join us and criticize us if we ignore some of the, significant development already out there. Thank you for the suggestion.
Awesome. Are there any other questions for Dr. Wang or any other
Like to
comments?
be fair, like I'm right, like me and Beth was trying to call for a working group doesn't mean like we know everything. We just aware of like maybe along with many of you as well, this important area, you know, your benefits if we work together. Doesn't mean we will know like every aspect answer. We would look forward the information, like more detailed guidance from you as well. It's just more open up for discussion. Like what would be the critical information or, you know, type of members we can recruit and join this force. And hopefully by the time this working group forms, then we'll have much more fun to dive into the individual like a topic and to be better, like as specific as possible. If we can tap a few, like, you know, Beth, you mentioned other amplification example through the working example, that can also amplify the necessity of such work.
Right, and I think that in between everybody on the call here and others that might be interested, we're going to have a lot of those working examples from a lot of different methodologies and just a lot of different perspectives on that, which is exactly what we need to build a decent data model. Great. So, you know, if we think that there's interest in this group at VICC, oh, Alex, I had another question for you actually. So is there a group at GA4GH that's already focused on kind of structural variant representation, or is that?
That's a good question. So there, the structural variation group, you know, there's an actively forming working group, and Wes, I think, is on the call and has been organizing this. But there's also the work that has recently been done over in VCF, which is also a GA4GH standard, to do like better phase groups and phase lists for structural variation representation in VCS. And, you know, I do think that there's a need for a, you know, scalable model that's applicable in a variety of scenarios. And, you know, that's kind of the purpose of the SV group that's forming. But, you know, there's, to date, I don't know of any other active efforts beyond those two.
Okay. Yeah, again, I was thinking if this was a topic at the GA4GH plenary, I didn't want to be, you know, proposing a, you know, directly parallel and competing group, but if we could collaborate with the people who are already working on it, I think that'd be wonderful. And you said Wes is there.
Yeah, and I think there are overlapping concerns, but this is not necessarily the same effort, right? So like there's, you know, in the VR like team, and the SV group there, the idea is really the minimum information around variation representation itself. But, you know, there's a pretty large discussion around all of the associated like clinical metadata, assay metadata, things like that, that are probably more of a broad clinically focused discussion that I think are maybe outside the purview of that very focused group over in GA4GH. So ideally, you know, there would be, you know, cross membership between the two groups so that what's happening in one informs what's happening in the other and vice versa.
That'd be awesome. Cool, thank you. Any other questions, comments?
For people that want to get interested, Beth, how should they reach out and let you know, or Dr. Wang?
You can reach out to us directly. I can share our emails, and then also if there is adequate interest here, we'll put together a project charter for VICC and send that through, and then hopefully get a project started here and work on starting to build up that collaboration with maybe a different group like CGC and some of those GA4GH groups, and make sure we have some crosstalk and membership there as well.
Yeah, and I think it's discussion between me and Beth that in the way we want to like set up some tangible goal, right? Because we know it's intimidating and it's a very hard topic. But if we can start something easy, just the way the different like aspect of participants will even reach out to the clinical lab who may consume the clinical structure variant or complex variant result. That will be like even focus on manuscript about the initial survey and the recommendation. It could be a tangible goal. And I'm enjoying the question and the comments so far, just hearing, for example, the metadata and the minimum information, as well as the ongoing work to clarify some of the terminology. These could be factored into the survey we send, like, I don't know, through CGC or the other clinical lab. Because on one hand, the research side, we are so moving forward to the long reads, and on the clinical side, fish, cytogenetic, and microSTM being used day to day to conclude the clinical diagnosis or some relevant event. So I think that's a significant topic. Require we start somewhere. Again, this is just to emphasize like our working group, even if to be formed, like we want to achieve tangible goal. Okay.
Really good point. Yeah, and typically our VICC working groups have been working toward, manuscripts or standards and guidelines, tangible efforts like that. So it's a good opportunity to, kind of gain some traction with that. Yeah, that's all I really have to say. Are there any other comments or questions? I put our emails in the chat, so you can let us know if there's any interest, and then we can put together a project charter and start to kind of connect people and different consortia groups as needed to move this forward. Great, hearing none, I'm thinking we might be able to end a little early today.
I just want to say that this is a very relevant topic, and I'm very excited that you guys are planning on starting a group around this to help advance the standardization in this space. Thanks for presenting, and I'm looking forward to what you two bring to the standards community.
Thanks, Alex. Yeah, I'm looking forward to it too. And I really think that, setting up this group kind of in an intelligent way and making sure that we're communicating with other groups and kind of using the information that other groups have already thoughtfully set up is going to be really integral to our success here. So I'm really excited to move forward and to work with anybody who is interested in complex variants. Oh, Cheryl.
Oh, hi. Yeah, I was just going to add, thank you also. And I think it might be worth thinking about separating the sort of copy number from the structural variants. I think both are really important to address, but they tend to be assessed differently, and they often overlap. Like, when do you call something a structural variant versus a copy number variant? And especially as groups are starting to assess more of the partial copy number variants, where it's not just whole gene gain, amplification loss, or arm length, but it's these focal or smaller even exon level gains and losses, that it gets really complicated. So it's worth, that's also worth exploring. But thank you for bringing this together. I'd be happy to work on it.
Yep, and Alex says ITDs, so internal tandem duplications. There's a lot going on there. And then also bucketed alterations, so which would be further downstream, I think, after some standards and guidelines are set around CNVs and SVs, we can start talking about how do we describe these really complex scenarios where we might have multiple pieces, multiple events in the same, or I guess, multiple alterations in the same genomic event.
Yeah, thank you for your comment. Just imagine there has to be like a figure one of ontology relationship, right? And the CMV and the structure error was like entangled, as you mentioned. But from the definition per se, I think a CMV copy number was triggered by the dosage change, right? But that dosage change could be focal, could be like a chromosome, like aneuploidy driven, mechanistic wise. But a copy number always associated some structure variant breaking point. And maybe it's always sub-branch of structure variants, just according to the observation, how we detect them initially.
Yeah, coming from cytogenetics background, it is news to me that copy number got dissociated from structural variant, because back in the days, in cytogenetics days, the division was, it is either whole chromosome gains and losses, and then it would be structural variants every time there is a breakpoint involved somewhere, and that was often copy number. That was all deletions and duplications. That's how we defined them back in the day, which means that most copy number was also considered structural. And now there is so much inconsistency in how those terms are being used that I would propose to this newly forming group to start by defining how do we want to use the term structural variant. In cytogenetics, structural is every time there is some breakpoint somewhere, and then we divide them into balanced and unbalanced. Balanced if there is no copy number change, unbalanced if there is. Now the usage of term structural variant is all over the place.
Yeah, thank you. That's a wonderful comment too. I feel like this should be like 3-dimensional or 4-dimensional ontology because depends on where you're coming from. But because some of the, I think, muddy situations caused by bioinformatics, right? Like we detect something and we call it the structure variant. And even further amplify, like even we have breaking point. I have some, like a colleague next to the door, he was a co-author on the like 1000 genome copy number structure variant paper, co-first author. I mean, they are also curious about and particular pay attention to the breaking point. Near the breaking point, once it happens, because the DNA repairing mechanism, there'll be homology or micro-homology, like footprint associated with the structure variant. So because you have such breaking points, then DNA repair machinery trying to compensate will introduce other error. So yeah, it can grow very complex, depends on where you're coming from. I feel there requires some convergence, at least for the representation, but depend on the school or technology where you detect the initial event or how you interpret the mechanism, what causes structure variant. There'll be like very complex research topic around them. And between me and Beth, we'll talk about like even the copy number burden, like overall copy number burden. Everybody talk about mutation burden as an indication for the immune response. But copy number burden, such as HRD score, homology recombination deficiency score, basically counting the number of large-scale copy number transition has an indication with a PAPA inhibitor, like target therapy. This is like before we can well define each of the events, there's already some accumulated score of copy number events used clinically. This go crazy.
Right. And I think in one of our recent-ish papers on, oh yes, I see that there's a comment. Yes, there will be a recording available for this. Yeah, in one of our recent publications, we actually did try to quantify copy number burden and then stratify it between copy number loss burden and copy number gain burden, just trying to look at all different types of complexity in acute myeloid leukemia genomes. So I can send that link around as well. So it's just a different way of thinking of things.
Yeah, with that. I think, if we want to be tangible, indeed, it would be nice and important to constrain our focus. But thank you. I largely agree that actually every copy number change should associate with some structure variant. like footprint, right? And maybe there's a companion like a micro homology or non-homology footprint. Depends on how deep we want to go. Yeah, and LOH. Yeah, thank you. These are all great questions. On the other hand, like numerous possibility. That's a good reminder we need to be realistic.
Yeah, I think we've established that there's a lot of work to do, which is good. That's the need is there.
Yeah, I heard like we have a lot of work to do, but I heard also repeatedly from several of you, like it's like the definition should be clarified, right? Should be a group of us with diverse background, but yet can agree upon. That's the same thing, whether we're using different language, cytogenetic or bioinformatic or like a genomic to describe the same thing. Maybe that's the first step. Like let's exchange the language and make sure we can converge on some like common ground.
I think that's really important for clinical reporting, that we know what we're talking about, that we get to a place where the, a bioinformatic detection of something results always in the same description clinically in a report.
Yeah, because bioinformatically to defend or like to clarify what we do, right? We write a program, you have FASTQ, we have BAM file, we see some change, we report, and we don't know necessarily how to report that correctly. We say, okay, structure variant. Then it's just adding the confusion to the field. So some guidance will be needed, like from domain expertise. Okay.
I think it's a really good point that definitions need to be established first, because yeah, I mean, even with the CGC discussion that we had, we kind of went rather quickly to, all right, well, what are we talking about? What are we talking about with complex variants, and what do we see as complex variants? And so kind of making it super clear what it is within our at least primary or preliminary scope is really important. So thank you.
Yeah, and thank you. And echo again, the definition is not only important as a guidance, because you can think about as engagement. As a bioinformatician, if I wake up, say, I want to develop another caller today, like I find another set of events, like I need a reference to guide me, like appropriately phrase and mention that type of event. So this definition will provide engaging guidance, hopefully for the bioinformatic research community as well. And we already have so many callers, like think about CMU SV caller. And I would think that could be a working example. I mean, by the time some of you truly interested in joining us, let's pick a few like popular caller, trying to match or gently modify that definition or clarify that definition. It will be super helpful for the bioinformatic field where I'm coming from. Okay. Yeah, I thought we can like end early, but thank you. This is a great question. But the important thing is join us,
Great. Well, yeah, it's kind of like there was something I read about, like the most important discoveries in the doctor's office are like the one last thing kind of discoveries right at the end of the appointment. So maybe this is kind of following that model. But thanks so much again for coming and talking to us. And we'll have the recording available, and we'll send out a link to the group, to the whole VICC group within a day or so. And yeah, you'll hear more from us in the near future regarding a project proposal.
Okay. Thank you, everyone. Thank you for your question. Bye-bye.
Bye.